to occur, the two pieces of DNA on that part of the chromosome move apart from each other. And you might wonder, well, how did that happen? Well, they're not held together super strongly. It's just hydrogen bonds that hold them together, and hydrogen bonds aren't super strong. And there's actually an enzyme that does it also, just like, well, I won't demonstrate, but think of your zipper. You have the two pieces together, and then that piece of metal in between can move them apart or bring them back together. There's an enzyme that can temporarily move the two DNA strands apart. Once the two DNA strands on the gene have separated from each other, there's another enzyme that comes in called RNA polymerase. Remember, all enzyme names end in ASE. That tells you you're talking about an enzyme. And the first part of the name generally tells you what the enzyme does or what it works with. So this enzyme's name tells you that it polymerizes RNA. It links together RNA nucleotides. And so what we're going to see happening is this. The RNA polymerase is going to latch on to this particular strand and ride along it like a train going down a train track. But as the RNA polymerase rides along that DNA strand, it's going to polymerize a new piece of RNA. It's going to make a new piece of RNA that's complementary to that strand of the DNA. Here it goes. If there's a T, it makes a A. If there's an A, it makes a U. If there's a C, it makes a G. If there's a T, it makes an A. That sort of thing. So you got a nice piece of RNA. It's complementary base pairing, T's with A's, A's with U's, C's with G's, G's with C's, that sort of thing. So notice that the RNA polymerase enzyme didn't have to know the sequence it was supposed to make ahead of time. All it had to do was just go along blindly doing base pairing when it makes its new piece of RNA. The rules of complementary base pairing allowed it to make that complementary strand without any prior knowledge of what it was supposed to be making. Now notice the codons that got made though. AUG, and look at this strand, ATG, GAA, GAA, CCC, CCC, UGA, TGA. So it's the same codons as this strand up here, but now they're written in RNA. And so notice how, I don't know, smart the engineering is on that. How did we get a copy of these codons down here? Well, because this new piece of RNA is complementary to this bottom strand, and this other DNA strand was also complementary to the bottom strand. And so since this and this are both complementary to the bottom strand, they must contain the same codons. They do, AUG, ATG, GGA, GGA, like that. All right, and that new piece of RNA that was made that has all the same codons as this strand of the gene is the messenger RNA, or the mRNA for short. And it's called that because it's going to send the message, it's going to send the genetic information, the codons, out of the nucleus to the ribosome. When the mRNA is complete, it detaches itself from that strand. There we go. Or maybe I should throw some terminology at you here. I didn't put this in the notes, but this is called the template strand, the one that the RNA polymerase enzyme rides along, making complementary RNA bases to, is the template strand. And the one up here that the RNA polymerase did not ride along is called the non-template strand. Yeah. I think I didn't hear everything you said, but they extract DNA from a what again?
Like from a shatter particle or from like spit. And then they get blood and they extract the DNA from that. And they put the DNA extracted from the hair of the spit into the blood.
Okay.
To frame somebody.
Oh, like from one of those CSI shows or something like that. I guess that might be possible, but the blood, your own blood has your own cells inside of it, so it would have your own DNA. And so if you put somebody else's DNA inside your blood, if they did a test on you, they would find 2 different sets of DNA. And so they'd know something was wrong because they'd say, this guy has 2 different sets of DNA, and every human being only has their own set of DNA. So I think that wouldn't work because you'd see twice as many genes inside the person when you tested them, and you'd know something was fishy.
Yeah, but what I said is that they extract the DNA from the blood.
God, they suck the DNA out of the blood and then put foreign DNA in.
Exactly.
I guess you could do that, but you have to burst open all the cells to get the DNA out of them. And so again, I think someone would say, if something's wrong with the blood sample, all the cells have been burst. And so that would probably be a giveaway. I'm not saying it can't be done, but I'm just saying, as far as I understand it, I don't know if you could get away with that. It's a clever idea, though. Okay, so where do we leave off? So the messenger RNA, this temporary disposable copy of one of the DNA strands, leaves the gene, exits through a hole in the nucleus, and goes out into the cytoplasm, and there it goes to the ribosome. And it's going to, the RNA is going to go inside the gap between the large unit and the small unit of the ribosome, and then using the codons in the mRNA, the ribosome will translate the mRNA, which means construct the protein out of it. And there's the ribosome. Here comes the messenger RNA with those codons, and so it matches them up with the appropriate amino acids, and that is how the protein gets made. Translation, when the ribosome constructs a protein from the amino acids using an mRNA as the instruction. Oh, just in case you're curious, what happens to the messenger RNA? Well, it'll float around for a while, and it's certainly possible for another ribosome to grab hold of it, so you might get more than one copy of that particular protein being made from the same messenger RNA. But eventually it'll be broken down. There are enzymes in the cytoplasm that purposefully will wipe out mRNAs so that they don't get stuck just floating around the cytoplasm forever. Good so far? Okay, there's one more type of molecule involved in this process that I didn't really show you so far, but I want to drag it into the picture now. Up to this point, when I've been showing you these amino acids that the ribosome has been building the proteins out of, I've just sort of showed them floating around inside the cytoplasm. And that's partially true, but in fact, each of those amino acids in the cytoplasm is being held by a special RNA molecule called a transfer RNA, or a tRNA. And they call them transfer RNAs because these tRNAs not only hold on to the individual amino acids, but they shuttle them to the ribosome. Here's what a transfer RNA looks like. And I put some RNA nucleotides on it just to remind you that it's actually constructed out of RNA. And at this end up here is where it holds the amino acid. Right there. So each tRNA molecule holds one particular amino acid in the cytoplasm and shuttles it to the ribosome. Now, before I leave this slide, there's one more thing I want to point out here. Those 3 nucleotides on the transfer RNA, at the very bottom of the transfer RNA, relate to the amino acid that it carries. Notice the codon for methionine amino acid is AUG. How does that relate to this sequence down here? This sequence is what to that sequence? Complementary, right. The complement of A in RNA language is U, the complement to U in RNA language is A, and the complement to G is C. And so this is what's called the anticodon of the methionine codon, which is AUG. And so for whatever amino acid a tRNA is holding, it will have the anticodon, not the codon for that amino acid, but the complement to that codon, the anticodon for that amino acid down there. Anticodon are 3 nucleotides that are complementary to that amino acid's codon. Okay, let's zoom back a little bit here. Oh yeah, I've got, I think, 2 more minutes, so hold tight for a second. So here's all the amino acids being held by their tRNAs. Here comes the messenger RNA, plugs into the ribosome, and now those tRNAs start shuttling their amino acids in. But notice something here, that the anticodon of each tRNA is what sits on top of the messenger RNA, like that, and here's why. That's what allows the amino acids to be brought into the proper order. Since the first codon is AUG, only a tRNA with the anticodon is going to stick to it by complementary base pairing. And so that's what ensures that the first amino acid that gets brought in is the right one for AUG. And the next codon is GAA, which stands for the amino acid glutamine. And so any tRNA that holds a glutamine will have the anticodon for that, CUU. And so that ensures that the next amino acid that gets brought in is the proper one that matches up with the GAA codon. And the next codon is CCC for the amino acid proline. And so the tRNA that holds proline will have GGG, the anticodon, so that it will stick to the next codon, and thus bring in the next amino acid, the correct amino acid, number 3. Isn't that clever? Are you impressed? It's amazing. You know, if you were trying to engineer something, you couldn't do better than this. It's just astounding. Okay, I think that's all we have time for today. I will see you all in laboratory. Bring your protein handout.